Welcome everyone. We'll get started here in a minute. Let people gather as we are here at noon in Central U.S. time. We'll begin shortly. We'll just give it a couple more minutes. Welcome everyone joining us. The numbers keep increasing. I wait for the numbers of attendees to level off here a little bit before we get started. Well, it's just about 2 minutes after. I think we can probably get going in respect of the people's time who have joined us already. Welcome to today's webinar and thanks for joining us. My name is Bill Cole. I'm a professor of epidemiology at the UTHealth School of Public Health at the Austin campus. And today's webinar, in a long series of webinars hosted by the Michael and Susan Dell Center for Healthy Living, is going to focus on current monkeypox outbreak around the world, some of the background about it and what people can do, I'm sure. Before we get started, I've got some housekeeping announcements. The webinar will be recorded, is being recorded right now, and archived. The slides and the slide deck, as well as the audio, will be found, and the video can be found on the Michael and Susan Dell Center website. If you go to msdcenter.org, give our experts 24 hours to clean it up and post it, that'd be great. I will be monitoring questions as the moderator during the presentation. If you put your questions in the chat box, I'll try to do my best to organize them. And when we have the Q&A session at the end of the presentation today, we'll be able to rely on those questions to start the discussion. I am so pleased, I'm always pleased, but I'm honored to introduce our speakers today, Dr. Susan Fisher-Hoch and Dr. Joe McCormick, who are both professors of epidemiology at our Brownsville campus, the School of Public Health at the Brownsville campus. Both have had a long and distinguished career in epidemiology, a variety of things, not just infectious disease, but have made a major impact on health around the world, starting with their training. Dr. Fisher-Hoch as an internal medicine specialist and a virologist, and Dr. McCormick as started off as a pediatrician and then moved on to CDC to have a long and distinguished career. We're just very lucky to have them both on our faculty. I think with monkeypox today, some of the numbers that we were looking at as of this morning, just over 18,400 cases have been reported since May 17th to the CDC in Atlanta. Interesting, I didn't know this. Monkeypox was originally discovered in 1958 when it was seen as an outbreak among clusters of primates used in research. The first human case was observed in 1970, so this is not a new virus. It is, however, modes of transmission and the outbreak we're seeing today are different. Previous outbreaks have been limited to Africa or have been linked to international travel to and from Africa or to imported animals. So things are changing here as they are. And we're just, again, delighted to host this webinar that will bring us all up to date on this. The title we're looking at today is The Age of Pandemics, First COVID and Now Monkeypox. So Sue, Joe, please take it away and help us understand this.
Good morning. I'm Sue Fisher-Hoch.
I'm Joe McCormick.
Now far off with, I thought you would like a slightly more photogenic copy of the COVID virus. This talk, in fact, is about COVID, and we will be talking about monkeypox later, as requested. And so let's go straight into it. If you thought you were done with COVID, I'll have to tell you it ain't done with you. And this is one of the problems. And we have a pandemic. Pandemics have been recorded for at least 5 million years. Call it what you will. 5 millennia. Pandemics and plagues, emerging viruses, call it what you will. Some have been emerging bacteria. And I just thought that you would like to just review the fact that this is not something new. And the common features in all these outbreaks are crowding and increased transport. Transport means movement of people and crowding of people. And you see, we go back 3,000 years. And here we are, 6 million and counting with COVID. And we haven't yet reached the sort of numbers that were seen in the Americas when the colonists first came over, or even the original plagues. So we've a little way to go, and it's probably because we can in fact manage it better now we have vaccines. So just to make sure you know what we're talking about, COVID is a coronavirus, and the coronavirus that causes COVID-19 is called the severe acute respiratory syndrome coronavirus 2, which is a mouthful. And so it's been helpfully shortened to SARS-CoV-2. Disease, however, is now you, the patient. And this is coronavirus infectious disease 2019, which is also shortened. These are animal viruses that jump from species to species, and they're very adept at this. And it belongs to an host of millions, maybe trillions of viruses in the environment that we eventually come into contact with. And once again, the pressure of overpopulation and crowding and civilization and going into the areas where these viruses were minding their own business is to some extent what has caused the problem with us. So the coronavirus, there are at least 36 that we know about, and probably a whole load we don't. There are 4 human coronaviruses. This is worth paying attention to, because these cause 1/3 of what you call the common cold. Just a plain cold in the head, and not usually with any other involvement of the lungs or anything. They occur in the winter. The problem with these 4 human coronaviruses, and we've known it for some time, is that classically immunity lasts less than a year, which means that the same virus can come back and catch you again. Maybe you've heard that story before, or maybe you've heard that story recently, but it is a pattern that coronaviruses clearly have developed. So what are the data? So as of this month, we've actually counted 391,000 new cases, or a little over in the last 24 hours. Cumulative cases are nearly 600 million. Cumulative deaths, 6.5 million. But the estimate, these are underestimates, and it has been put forward that they may in fact be 10 times too small. So the numbers we're actually looking at may not be 6 million, but 60 million deaths. We don't know, because in so many places they're not reported. So these underreporting is a big issue. Here is the pattern of coronavirus reports across the world: Europe, America, Western Pacific. You see, Europe has got by far the most. The Americas has got a lot, but we get down here to Africa, hardly any. That's probably where the underreporting really kicks in, that the systems there for reporting cases, for testing and reporting cases, are pretty scant. So the global situation and the deaths, as you can see here, here is the pattern over time. This was the Delta, and this is the Omicron surges. And here are the deaths. The important thing to understand here is the deaths here in the earlier years were much, much, much greater, and we seem to have controlled the mortality, if not the actual epidemiology of the disease. So what's been going on? So the story is the variants. And what happens is that coronaviruses, like many viruses, mutate. They mutate in the face of opportunity. So this means that a very high rate of infection of these viruses means billions and trillions of new virus replication events may be occurring every day. So and these are very high. One person had up to 10 to the 11 RNA copies and 10 to the 7 virus particles at the peak of infection. That is a lot of virus. And as the virus replicates, it makes mistakes. It's an RNA virus, and RNA does not have an editing process, which DNA has. Your DNA in your cells has an editing process, which picks up mistakes made in replication or cell separation or cell reproduction and corrects it. The viruses do not have this system, so they make all these mistakes. But what the virus does, it takes advantage of these mistakes. And most of them can be fatal to the virus, but there'll be one or two that actually give the virus a big advantage. And these are the ones that overtake and replace the other. That is the process of a variant evolution.
Let me just add to that when you look at one person that may have somewhere around 10 billion to 100 billion RNA copies during their infection, at the peak of their infection, and they may have 10 million to or over infectious virus particles. You can see what the odds are of since they all involve some level of mutation. You can see what the odds are for eventually having variants that emerge from all of this and give the virus eventually an advantage. So this is the virus's great evolutionary advantage. It can just mutate all over the place until something emerges that makes it more infectious and not necessarily more severe, but maybe.
So this is classical evolution. If you don't believe in evolution, take a close look at this virus because it's doing it fast forward. So why is it so successful? Because human factors actually help the virus. And unvaccinated individuals give it a big advantage. Vaccine refusal, obviously. High rates of infection in crowded and disadvantaged places. And this happens all over the world. So many of the emergent strains have come from places which are very disadvantaged and highly crowded, such as South Africa or India, and sometimes in South America. Uncontrolled large gatherings like the one you see depicted below on the Ganges is a great place for the virus. Failure to wear masks, failure to wear them correctly. How many masks have I seen below people's noses? I wonder why they bother. And then also there's this thing that's really emerged more recently, persistence of virus in the immunosuppressed, such as people with HIV. It can persist, and as it persists in the individual, it can mutate. This may also be true in people who are taking immunosuppressants. There are a lot more people now on immunosuppressants, and they are particularly at risk from this virus. So the variants, to try to track this, and an effort was made to name them by the Greek alphabet. We got more than halfway down to Omicron, and we've kind of given up. So the red ones are the variants of concern. These are the ones that have really caused a lot of deaths and a lot of illnesses. Alpha, beta, gamma, delta. It was going fine up to delta. Then when we started to go down epsilon, zeta, theta, we got into trouble until we got to Omicron. And the Omicron is a new lineage, which means it actually re-emerged in a totally new form. And the BA5 series, which you may or may not have heard about, because that's the one that's circulating recently, they seem to have given up on going down the rest of the alphabet. They were running out of names anyway. Anyway, the problem with the Omicron variants is that they're increasingly more transmissible. That's the advantage. But they seem to be less virulent. And that is a very good thing for us. And this, in fact, is an advantage for the virus, because if it kills off its host, it dies with the host. But if it can keep the host alive and get them transmitting, it does much better. So you can see here that the Omicron has chosen a path where it can transmit and doesn't necessarily kill. Let's just quick look at these variations. And these are the number of mutations beta, alpha, delta, gamma, and then Omicron has all these new mutations in the spike. The spike, I'm sure you've heard about, it's on the surface and it's made up of 3 identical proteins. It's sort of wrapped up a bit like a cauliflower head. And each stalk has 3 vital regions. Receptor binding domain, that's the one that actually attaches to the cell and gets in. And then the N-terminal domain and the furin cleavage site. And this is where the mutations are. So here's a picture of its sort of cauliflower head and the number of Omicron mutations. And the first point of the mutation that's really important is the receptor binding site. It acts like a key to open the cell. And you see that Omicron, all these dark orange ones, are many more mutations, whoops, in that site than there were in the other ones. Back to, sorry, doesn't go back as the way it should. And you see, these are the ones that were actually the same, and then there were new ones. So it's got a lot of mutations. It's got a lot of mutations in the immunity site, which is the one that where you actually protect yourself, where your cells attach and try to kill it. Here again, you see all the mutations it's got. It's also got mutations, but not so many, in the severe illness side. And I think that that is where the virus has chosen the most advantageous mutations, or the mutations have chosen the ones that will actually replicate. So we'll just go quickly on to surveillance, and I'll pass it over to Dr. McCormick. We've been using testing for surveillance of this vaccine, and it just stopped working. It doesn't work for a lot of reasons. People don't get tested. You saw the international data and how very low numbers they have in countries that have poor testing facilities, and therefore you just don't get the numbers. Poor and remote communities are overlooked. People don't come forward for testing. Now that home testing and the home kits have replaced the testing centers, so we don't, with those people are not reported. Asymptomatic cases won't get tested, so you won't see those either. And those people who die at home will be missed. So that leaves us with a problem. It means that the data we have right now for surveillance is inadequate and very, very patchy. So there is a new tool.
Let me just make a comment about that, because I think there's another important issue here. So one of the things that's become very clear is that the vaccine continues, even though the virus escapes, as Dr. Fisher-Hoch just showed you, with some mutations. The vaccines still work, but people can get infected with the virus even if they're vaccinated, and now we know people may get infected multiple times. They probably aren't mostly very ill and so they may test at home. They may not go in for testing at all, and it doesn't get reported. So it means that our surveillance is even less effective. The good news is vaccines work, and the not so good news is it complicates our surveillance because people are less ill. They don't try to get treated, are tested. And so we have much less insight into the spread of the virus. But the good news is we don't see the hospitalizations and the severe disease.
So there is a cool new tool. This is testing in wastewater. Turns out that most viruses are excreted in the feces. And so, like it or not, that's going to get into the wastewater. And so this is really a method for universal testing, not individual testing, just universal testing. And here is from CDC, is a cartoon of how it works. And this is particularly, I think, from San Diego. I'll show you the data in a minute, as you can actually test at the university level, and in a residential neighborhood, or at a treatment facility. Most people are now doing it at the treatment facility, which gives you, in fact, quite a heads-up analysis, because the actual technique you use is relatively simple. So you get the wastewater, just a small tube of it, and you pasteurize it so that it's no longer got infectious particles. And then in the lab, you run it through a PCR system, which is exactly the same as the testing from nasal swabs. And it detects the virus really in very low quantities, extremely sensitive technique. So the CDC has set up a national wastewater surveillance system. And this wastewater contains genetic material from viruses, bacteria, which can be captured by PCR. And these studies, including New York, which New York's been doing it since 2020, so have places in California and some other places. We're in Colorado now, they've been doing it here for a long time. And this, in fact, gives you a 10 to 12 day advance notice of a spike. So this is really helpful. You don't wait for it to happen. You get advance notice that it's going to spike. And this has really helped with prevention and mitigation systems. So as you can see, it's really been useful. However, if you're sitting in Texas, you're not doing very well, because you can see that it's spotty at present about who is actually collaborating with the CDC. You will see that here in the Northeast and in California, they're doing quite well. They're doing pretty well in Colorado. Texas is pretty pitiful. But I would like to tell you that we are doing it in Brownsville. We're doing it in Tyler. We're doing it in El Paso. And Rice has been doing it in Houston. So something is happening. And I think this will grow, because in a minute I'll show you the adaptation. So this is one example of the University of California, San Diego campus, the larger San Diego. So they were able to test each dormitories as well as the general effluent that came out. And they got the Omicron variant up 10 days before the country's first clinical test. So it really is an early warning system. And they were able to show when the BA1 Omicron took over from Delta in late 2021. And so it's really taken off. One other very interesting about it, on the right, you'll see the new data from New York. And here they show which they were. They were able to sequence them. So here is iota, alpha, beta, and gamma, delta, epsilon, other, unknown. So you may say, well, I wonder what that is. And they wondered what that is too. These particular isolates in the wastewater did not match any human samples. So there's some question that maybe an animal reservoir, possibly rodents. Excuse me. So this system also detected poliovirus, which just shows you just how flexible this system can be in epidemics. And this sequence was identical to an active polio case. You may have heard of this, probably imported, a person who had received type 2 vaccine, which managed to revert. And this is the oral polio vaccine. We do not use the oral polio vaccine in the US. We use the intramuscular vaccine, which cannot mutate like this. And this particular case had paralytic polio. So this was another extraordinary use. Now we know it can detect monkeypox. So Joe, do you want to take over the long haul? I think I've talked enough.
So let's talk just a moment about the other feature of COVID that is emerging on the scene, and now we've all, I think, heard a lot about it, and that is the long haul syndrome. And it is a rather vague definition because it includes so many different things. So it's defined kind of as a wide range of symptoms that can last more than 4 weeks or even months after infection. As you can tell from the definition, is not well understood. We're still learning about what some of the long-term symptoms can be, but common signs and symptoms that can persist depending on the individual. It varies from individual to individual. Fatigue, shortness of breath, muscle pain. Really, sometimes people complain of brain fog, memory, concentration, and sleeping problems. Loss of smell and taste, which is already well known, and many other, including changes in their heart rate, a lot of other things that can, and of course that means the heart, the lungs, the brain, and even the vascular system can all be affected long-term by this long-haul syndrome.
Just one comment about it is may affect as many as 1/3 of people who have COVID, symptomatic or unsymptomatic. And it's interesting, it also resembles this type of syndrome we've seen after the 1918 flu epidemic, and also the one in 1889.
So yeah, we're seeing and here, this is an example of one of the studies done. There is a very large database in the United Kingdom called the UK Biobank, and it's about 500,000 people who have been very carefully characterized over the years. And one of the things because they had prior data before COVID, they were able to go back and look at a particular study, and this is a study looking at changes in brain structure in people in the UK Biobank. And what they were able to do is take 401 cases of people who were infected with SARS-CoV-2 between March 2020 and April 2021. Only about 4% of these people were hospitalized. They had a set of matched controls, that is, people who didn't get infected. And they compared then they had brain MRIs on these people, and they compared the cases and controls. Well, what they found was among the cases, there was a loss of gray matter thickness and tissue damage in areas associated with sense of smell. And remember, one of the prime symptoms of many people, particularly early on with COVID, was loss of smell. So here is some primary evidence that in fact, there was damage in this area. The whole brain volume was shown to decrease in people with COVID. And there was a decline. There were a number of tests being done that reflect people's ability to perform complex tasks. And there was a decline in this ability. So what we learned from this is that marked changes can occur with the olfactory pathway, that is a sense of smell and taste. These changes increase with age. We don't know if they are reversible, but we know that people's cognition is affected. And of course, we need to do more studies of this. We happen to have such a study, much smaller. We don't have this kind of numbers, but it was pretty impressive. And I think it really propels us to better understand what the post-COVID long-haul syndrome is really going to look like, not just weeks and months, but even years later. The other area that is of considerable importance in the long-haul syndrome is the impact on the heart and the cardiovascular systems. And the number of people, and this was a paper just published this year, showing that among 1,000 people with COVID, 45 out of 1,000 had some kind of cardiovascular outcome. Major events, at 23 out of 1,000 had strokes. 19, almost 20% had abnormal heart rhythms. A smaller number had what's called atrial fibrillation, which has become a very prominent thing, particularly in our elderly population. And other things like strokes and heart failure occurred in as many as 10 people per 1,000. So this is clearly important. That means one person per 100, which clearly shows that we have a lot to learn still in terms of the long-haul syndrome, and that's something we're focused on. The good news about all this has been the impact of vaccines that really has made an impact on the hospitalization rate and death rate as more and more people have gotten vaccinated. Indeed, if you see today, if you look at it, we'll show you a graphic a little bit later, showing you what happens when people are unvaccinated versus vaccinated in terms of hospitalization and mortality. Here are all the vaccines, just a list of all the vaccines that are available throughout the world. In our area, Pfizer, Moderna, Johnson & Johnson are known. We now have a new one that's been approved called Novavax. Novavax has also been approved in the European Union, Indonesia, and the Philippines. And its advantages that it's a protein vaccine, which means that it can be transported and stored much less with much less complexity compared to the very low temperatures that we have to keep the mRNA vaccines. So for areas where these facilities for conserving the mRNA vaccines are not well established, this can be a real boon. Just to remind you that, again, this is what our DNA looks like. We make what we call messenger RNA off of our DNA, and it then eventually hooks onto a ribosome and makes protein. So the vaccines include this level of RNA. This is where we have Moderna and the Pfizer. And then this is where the Novavax vaccine occurs. It's actually when the protein itself. So we go way down from DNA vaccines all the way down here. Now the J&J vaccine is in fact a DNA vaccine because it's the DNA, the same DNA that reflects the mRNA, but it's put into a vector called an adenovirus, and it goes through the same process to eventually make mRNA. These are the two people who did the outstanding work that led to the mRNA vaccines. They are located at the University of Pennsylvania, and their work really provided the breakthrough that was required to be able to make injectable mRNA vaccines. This is the graphic I promised you. In New York, this is what happens. This is hospitalization of people who are unvaccinated compared to vaccinated. So if anybody ever needed persuasion that maybe it's a good thing to get vaccine, this might be, if they're persuadable, that might be one of the things that would persuade them. This also suggests that about 234,000, this is from the CDC, suggests that about 234,000 COVID-19 deaths since June 2021 could have been prevented with vaccinations. Almost all of these people refused or did not get vaccinations for one reason or another. You want to?
Yes, I would say that to this morning, the FDA has announced that a new vaccine will be available. It's been shipped out this week. Millions of doses will be shipped out this week. This new configuration now has the BA.5 Omicron in it, as well as the original Wuhan strain. And here is the statement from Moderna's CEO, who's Stefan Bancel. He says it's compared it to the iPhone. He says we'll need a new one every year. And I think we'll have to get used to that. But fortunately, the mRNA technology is a bit like typing in a new message on your computer. You just have to change the formulation and you can do this sort of very simply in order to make a new vaccine. So we're in a very good shape here. So updating the vaccines is going to be the deal. So off you go, everybody. Tomorrow's the week to go get your flu and your updated COVID vaccine, and you'll be good for the winter. So the evidence for efficacy for booster shots, do you want to talk about that, Joe?
Yeah, first of all, the booster shots have improved dramatically protection for immunocompromised. Who are they? People with autoimmune disease who are on immune suppressant drugs, people with cancer who are being on immune suppressed drugs. So we have many such people now. Because of modern technology, people are surviving but being treated with these suppressant drugs, and including rheumatoid arthritis and eczema and many other conditions like this. But they are more susceptible to viruses like COVID, and so, but the vaccines do work well in those people. It obviously helps prevent hospitalizations and deaths. We know that all of these vaccines work, some a little better than others, but they all, in the overall conclusion, vaccination with any of the vaccines reduces hospitalization and death. Let's talk for just a moment about treatment. Many of you have heard about Paxlovid now, and the fact that it is a drug that, when given early, can reduce the level of hospitalization death, but it needs to be given within about 5 days of onset, which means people need to get tested, which has now become a little bit of a barrier. We also know, unfortunately, that there can be a rebound. We know the president had a rebound when he was treated with Paxlovid. The rebound in the sense that it reduced the virus for a while and then it rebounded. There are a bunch of other drugs out here that are under trial in various ways that yet are not yet FDA approved. And then there's a lot of stuff that doesn't work, like ivermectin and hydroxychloroquine and a whole bunch of others that don't work. How do these work? Well, the monoclonal antibodies are ones that you've already heard attach to the virus and prevent it from getting into the cell. That's also what our vaccine antibodies do, the same concept. The drugs, on the other hand, get into the cell and prevent, they actually interfere with the virus replication. I won't go into the details of how they do that, but a lot of them, and they attack it in different places. Here you can see that a lot of these drugs work here, or here, or here, or here. So the fact that we understand the replication of the virus so well means that we can go in and develop targeted drugs. Now, you've heard of dexamethasone that's used widely, and that is one that has a much broader impact on the immune response. And basically, it helps to reduce the immune response. The immune response is partly responsible for some of the symptoms we see. So it's almost an overreaction to the virus infection.
I can make a comment on that. You don't want to give the dexamethasone too early because you need your immune response to the virus when it first attacks you. And when it's gone away and leaves this cytokine storm, that's when you need the dexamethasone. One comment about Paxlovid, it has occurred to me since it inhibits replication, doesn't actually kill the virus, which may be why you get the rebound when you stop.
Right. So we'll talk a little bit about mitigation, but I think everyone knows that wearing masks is effective. And this is an illustration of where, in places where masks were used, such as California, New Jersey, and New York.
But look at the death rate up there in New York, that blue line.
Yeah.
And it's right down here now.
When they started using masks and
you see that Florida, Georgia, and Texas have earned gold stars
for not using masks and not getting vaccinated.
Yeah.
So we know the key to spread is being crowded, poorly ventilated areas. So when you go into such an area, it's still a good idea to wear a mask. I'm going to jump over this now so that we can move on into monkeypox and spend a few minutes talking about monkeypox.
Just introduce it, the virology. It's a member of the family Poxviridae, the genus orthopox virus. That doesn't mean much to you, except that orthopox viruses include smallpox, which everybody's heard about, which is the variola virus, the cowpox virus. It is not related to chickenpox. It's got nothing to do with chickenpox. That's an entirely different virus. It's a DNA virus, not an mRNA virus. There are two strains that we know about, originating mostly, as far as we know, uniquely in Africa, in Central Africa and West Africa. It was first identified in 1958 in a monkey colony, so it was obviously an infection in a laboratory facility with monkeys. And it was thought at that stage to be a virus endemic in African rodents. The only outbreak that's ever been in the US was in 2003, and it was the West African strain, and it was in US imported pet animals, and people very closely associated with those animals. I do emphasize closely associated. So here is the whole orthopox virus species. You see, smallpox is way up there, and the monkeypox strain is way down here. Come down here, Jeff. Right. Monkeypox. So it is very distantly related to smallpox. Vaccinia is the vaccine that we use. We're not quite sure what the origin of that is. It may have come from cowpox. We don't know. But it certainly has been a very effective vaccine against smallpox, used usually in ring vaccination, which means you vaccinate around a case. The nice thing about smallpox is you never miss a case because you always see it. And therefore, it's possible to do this. Go on.
So it's a zoonotic disease. People get infected through direct contact with animals, and also it's transmitted person to person through direct close contact. This is not an airborne virus. There's no evidence that it spreads in any way similar to COVID, for example. We've had, as was mentioned, outbreaks in monkeys and also domesticated prairie dogs in the United States. Again, after being exposed to animals from West Africa. Similar to smallpox, it does cause a rash, and it's often identifiable through this rash, but not everybody gets the rash, as an exception to smallpox.
But it is excreted in feces, so we can apply the wastewater system to it.
Now, monkeypox virus appears to be able to infect a wide range of mammalian species, including dogs. So the jury's still out about what's going to happen with this if it gets into some animals here in this country or in other countries. The monkeypox virus now is being spread in many different countries from person to person. We know less about what it might happen to it when it gets into animals. It's known to be to infect a lot of different mammals in West and Central Africa. And just to give you a bit of history, yours truly, standing there with a beard, was part of a monkeypox field team in the Democratic Republic of the Congo around 1980, when we were trying to understand the extent of transmission. Remember, this was only a few years after smallpox was eradicated. And this team, we were trying to understand how much transmission is there in West Africa with this virus. And basically, we only found about 5 to 7 generations of transmission before it sort of died out. And that's rather much what we're seeing now today in this country. So it was declared as a public health emergency July 23rd. This year, the first case in the US was in May. And as of August 9th, 32,000 confirmed cases across 82 countries. And now I think Dr. Cole reported many, many more cases now in the US. It's transmitted directly skin-to-skin contact. And in this outbreak, in our outbreak, mainly among men with intimate sexual contact, it also may be able to spread through fomites, clothing, sex toys, bedding. The mean incubation period is estimated at 7 days. We don't have a lot of good data because it's a relatively mild disease, so people are not seen unless they get really get a pretty extensive rash. Here's the global spread right now of what we understand. This is all the countries where we're seeing this in humans. And in the US, this is our map showing you, and you can see that Texas is up there with 101 to 500 cases, but of course Texas has over 25 million people, so there are a lot more people to get infected. California, see some of the more populated states, are the ones that have the most cases. People are ramping up surveillance, and now with the potential for wastewater surveillance, this is going to improve. This shows you confirmed monkeypox cases in the US through about the middle of August. And you can see now that it certainly is increasing in its numbers. This is a rarely fatal disease. We don't, as I indicated earlier, we don't know a lot about what's going to happen if it gets into people who are immunocompromised. But at this point, it's a relatively mild disease. People may have flu-like symptoms with a headache, fatigue, and malaise. Rash does appear almost always, but not always. It starts with a little red spot called a macule, and then it can become raised and like a blister or a vesicle, and then with even pustules. So there is actually, they get infected locally and can become a little pustule. In terms of, there is some pain associated with the skin rash. People may also have a sore throat and rarely have something called epiglottitis, which is a swelling of the voice box in your throat. And tonsillitis has also been. Rarely pneumonia, encephalitis, and eye infections. They have been seen in young children. And as I said, the impact on immunocompromised is not known. Treatment is mainly symptomatic. There is a drug that's not FDA approved, only covered by investigational new drug type of approval. It's in short supply, and it's only really available for high-risk patients, such as those people who are immunocompromised. There are several clinical trials that are underway, but it's still early days, and we don't know much about it. It again is a drug that has an impact on the replication of the virus. But we still have much to learn about this drug. And as I said, it's not FDA approved yet, even though there are clinical trials ongoing. And it remains to be seen how effective it is, and two, whether it will be licensed.
So protection really is about your own behavior, limiting numbers of sexual partners, and that includes kissing. Any kind of intimate contact will spread it, and avoid those places where this may occur. Condoms have been recommended, but they will not prevent all exposure, because the lesions can be elsewhere on the body. And washing certainly helps and take care of fomites, which is any kind of bedding or other thing. The vaccines that are available are not FDA approved. The vaccinia virus, which has been used for smallpox, is available for smallpox, but it's called Gymneos, which I cannot pronounce. And it is, you have to be very careful with the current attitude to vaccines, because this vaccinia virus does have side effects and has some serious contraindications, for instance, pregnant women. There are some other possibilities. Whether or not we will have a vaccination campaign, I think, remains an open question.
Again, one of the things that might happen is that we may elect to do ring vaccination around cases. That's also potential, but even that is not decided yet.
So here is the, you vaccinate first, but as soon as the infectious cases, you get the antiviral treatment. So this is the kind of pattern that you would expect for management of the disease.
If you get the vaccine, and most people won't get it, it takes 14 days for it to be effective. So we'll stop there and open it up for questions.
Fantastic. Thank you both. I was struck particularly by some of your first words, Sue, as we began the hour, talking about crowding and talking about overpopulation and so forth. Is this the future? One new or one dominant outbreak after another as we get more urbanized, as population increases and so forth? Are we looking at a never-ending cycle of outbreaks and pandemics?
Yes, and the reason for this is because of overpopulation, and viruses and bacteria too are opportunists, and we give them the opportunity. So yes, that is in fact the case. In fact, you can see it also in some of the things we've done to the landscape around here, where we also live. There has been a disease called of the pine forests, and it's due to monoculture. We did clear cutting and came back with just one species, and so a fungus got in and really ran riot with that species. We're doing the same thing on the human scale. We are replacing all sorts of other species and becoming a kind of monoculture, and that is a beautiful breeding ground for pathogens.
So many years ago, when Sue and I were working on Ebola and Lassa, and wrote a book about our adventures, one of the things we pointed out was, in the world of viruses, we are the invaders. And I think what that suggests today is that the fact that we have so many people, we're moving into environments that we haven't been in before, and these environments contain viruses that we haven't even recognized. But they have that potential then, just like the original SARS and now the COVID coronavirus and others, such as Ebola. now have moved in and can start to create pandemics in our population.
So really, what we're doing is we're laying out a feast for pathogens.
And with over 7 billion people,
I was struck by the frequency of replication. You get that much replication, there's bound to be an error, and and then we're off and running with this. I've got a couple of that's great. Got a couple of questions from the audience today. Don't know who these people are, but I see them here at this stage. Let's take a scenario of someone had never gotten the original 2 vaccines for COVID-19. Can they still begin the process of vaccination to help prevent the disease? Is it still effective? Should they bounce right to the booster? I guess this kind of gets at the difference between the base vaccine and the boosters.
Yes, you should take the booster. And the reason that this is it also contains the original vaccine. So it gives you, 2 for 1. And anybody who's not vaccinated should most definitely get vaccinated.
You saw the graphic of vaccinated versus unvaccinated for hospitalizations, and of course that translates to deaths. Why would anybody not choose to get vaccine? These vaccines have been given to tens of millions, hundreds of millions of people with almost no side effects. You don't know what else to say to people. It's a safe vaccine, and it could prevent you from a serious illness.
These may be some of the, if not the safest vaccines we've ever produced. In the past, vaccines have had other components in them which give us problems. And as I said, the vaccinia virus is one to be a little bit leery about, but these mRNA viruses are really, really clean.
Just to be clear, is it your recommendation that someone who hasn't been vaccinated up until now and wishes to, they should start with the original 2 full doses and then move to a booster, or should they just begin with a booster?
No, they would go into a full dose regimen, but with the new vaccine.
The new vaccine.
The new vaccine starting today. Mm-hmm. Right, because that covers it's bivalent, I guess, right now. Great. Let's say another scenario has popped up here. Someone who might have gotten a second booster shot last week. How long should they wait to get the new bivalent vaccine?
This morning's recommendations are 2 months. 2
months after, at least a minimum of 2 months after getting the second booster. Okay. Let's pivot here. We got about 6 minutes left. A couple of questions on monkeypox regarding testing. How available is testing for a monkeypox infection? Is it at any ER doc in the box? Is there a special protocol to have to be tested?
No.
What's the status of your understanding of testing right now?
No, testing is a problem because the only test available is not FDA approved. It comes from CDC, and they're only sending it to CDC approved laboratories. There are a number of them across the country. So you can get tested, but you're absolutely restricted to that. There is no commercial kit available. I believe people are working on it, and no FDA approved tests.
It is a PCR test at this point.
And it's not from a swab, it's a swab from the actual vesicle, the actual lesion on the skin.
Okay, so not nearly as available, obviously, as where we are with COVID-19 right now. I have to. I was sitting here smirking because I turned the camera off. I caught COVID finally this summer and did home testing, and I never reported it. So I'm part of the underreporting problem.
There you go.
Right.
Back to monkeypox. You talked about what we know so far, Joe, about transmission. It's not exclusively a sexually transmitted disease, however. What I've been carrying on. Do you have any idea about the dominant source right now of transmission? Is it largely sexually transmitted, or is it multiple ways?
So it's close contact with somebody who's infected and has lesions, And it is not, and that while most of that is related to sexual contact, it's not necessarily that. There are other any kind of close contact with someone who has lesions can be can transmit the virus, to be sure. So, you know, but it has emerged similar to what HIV did early on as predominance of people of men who have sex with men.
And it's not like smallpox was actually transmitted by aerosol, but monkeypox apparently is not. So that's one of the reasons why it's far less fearsome.
I did see a recent report in an MMWR report about fomite testing in a household with two members who had two members of the household had monkeypox. It was hard to find living virus on countertops or towels or anything. So fomites may be a secondary transmission mechanism.
It's a much more robust. These double-stranded DNA viruses are much more robust than the COVID virus, which is RNA and is relatively fragile. So it does persist. But it was very interesting, that article really, PCR will pick up genomic fragments, but what they could not do was actually get live via virus, which they could cultivate out of those specimens. There may have been some there, so you never quite know.
And it's live virus that's needed for transmission.
Correct. Terrific. I got one more sitting here, and I'm glad we have physicians in charge here. What is your opinion of the intradermal method of administering a monkeypox vaccine? I guess rather than intramuscular.
So intradermal is well known to be a very effective way of delivering some vaccines. Rabies is a good example of that discovered a long time ago that that giving intradermal rabies vaccine was very effective, and it reduced the dose that you have to give. And that's been the advantage of the monkeypox. Clearly it works. And what the intradermal route allows is much lower dose of the vaccine, which is, as CDC pointed out, expanded pretty dramatically the availability of vaccine.
Yeah. Another question popped up. You alluded to it nicely, I think. In addition to the mRNA vaccines and the rapidity that we've built on the last 20 years of research in that area to get the vaccines out in record time, I think one of the biggest steps forward, at least in public health surveillance, is in fact the wastewater tools that we're using. If you can find a canary in the coal mine and have actionable steps 10 days before you can pick up any evidence of an outbreak, then it seems to be a great early warning system. A question popped up here: how can we improve wastewater testing in the state? You had a couple of dots around various large cities, but I didn't see any in rural Texas, for example. What is your thought about doing things differently? What can we learn from California and New Jersey and those places?
Well, Texas the state declined to join the wastewater surveillance system. So it has to be up to people like us to take the initiative and to move forward. And that's what's happening, but only in some places. For instance, this week we have been sampling, believe it or not, on South Padre Island. Very important place for us to look because we have so many visitors coming in and out, and therefore transmission is likely. We're starting with Brownsville. In El Paso, they've been testing for some time, and in Tyler, they've also been testing for some time. But it has depended on individual people taking initiative and setting up the test, and that's what we're doing.
And making it more widespread, making it more available. I mean, one of the issues for a long time with these pandemics has been the availability of laboratory testing well out into the population. If you recall, when we had H1N1 about over 10 years ago, it was all started with depending on sending specimens to CDC. Well, that was just a non-starter because they had no way to test all the material. And even at the state level, for most of this, the state, for all of the states, the state laboratories alone don't have that capacity. Now with the technology we have, we can expand the availability of laboratories, but it requires some investment and an incorporation of existing laboratories into the network of laboratories that are available for doing this kind of work, and that includes wastewater testing.
And the initiative in our area is clearly very interesting because there is a coalition of the county and some of the cities who got together and agreed that they needed better support for epidemic surveillance and management. And we have been played a very big part in that. And one section of that was to help us get our laboratory up to speed with FDA-approved testing and other more sophisticated things, which is why we're now able to do the wastewater testing for our area, for Cameron County. It's not available anywhere else in South Texas, but it was initiative not just of us, but of the city of Brownsville, Cameron County, city of South Padre Island, other places said yes, and they actually put up some money to make it happen. So money is key.
Partnerships and collaboration, so the essence of public health, I guess. Doctors, thank you both very much. We're a little bit over time, but I wanted to thank you on behalf of the Michael and Susan Dell Center and all of our participants for sharing the updates and the expertise that you always bring to these. For attendees, the recording, the video and audio, as well as the slide deck will be posted within 24 hours, I hope, on the Michael and Susan Dell Center website. And until next time, thank you all very much. Have a great day.
Thank you. Our pleasure.